right, thank you. So before we get into the R-code stuff, I thought I'd bring up the BigOmics software since there were some questions about it yesterday. And so what this software will allow you to do is actually load in your raw data. And then it does all of the differential expressions. So here we're looking at just a TMT example of a data set. It kind of shows you the overall expression. It says genes, but this is protein data that's listed. It has all of your clustering features, so you can cluster by samples, you can cluster by features, different factors. You can look at filtering out different samples, cell cycle, all kinds of different stuff. But then it also has all of the differential expression, volcano plots. They actually run multiple analysis methods in here, not just one. So this one's got Lima-Boom, Wald's test, EdgeR, some different options. What's nice about that is in the table, you would actually find proteins and they have a star if it's found in all of the methods or not. So you kind of get this consensus of what proteins are going to be important regardless of what method we're throwing at it. So that's a really nice feature. Also has some correlation analysis, just looking at various functions and how it's related to different things. Let's see. The one that I like is also if you have biomarker studies that you're looking for, it will, you can compute biomarker analysis on here, which I didn't run yet. But you have the gene set, so gene set enrichment with all your pathways and your functional drugs that are related in this particular data set. So connections to drug pathways, ontology pathways, all sorts of other pathways. Adding people to the Zoom. So pathway analysis is going to have Reactome, wiki pathways, other type Go graphs are connected as well, and you can zoom in on all of these plots. But the comparison data set for like some of the multiomics stuff is fun because then you can load in, say, your protein data set and your RNA-seq data set and actually start to do a comparison within the software between what proteins are here. So there's a lot that I can't cover in here because it's like when I met with the guy, I went over all the different features. That was a 2-hour meeting. We don't have a 2-hour meeting. So there's quite a lot of power within this software. So, and they're continuing to build more. It is a fairly new, young company. So they're really happy that I'm giving them free advertisement. So just wanted to mention that one.
What sort of data?
So this will be count data for RNA-seq, any omics type data set on the genomic side, but the protein data I loaded in my normalized intensity file. So they have a normalization in the software, but again, I just turn that off and go ahead and normalize my data set myself and then load it into the software. So a lot of people asked them for that, so they went and added that feature. Anything else on this one?
Stephanie, what do you have to use as the identifier?
UniProt, they've
Yep,
added UniProt IDs.
Okay.
And the other thing is the organism. That's always a popular question. So they have over 200 species that are currently in there. They are working on the bacterial side. That's not quite there yet, but they're adding to the organism list too. So yeah, it's as I learn more, as I get into this deeper with these different projects, I'm going to try to break a lot of these different modules and steps again into another little video. And I'm trying to keep my videos short so they're not just like hours of listening to YouTube and me talk. But it's going to be helpful because here you can actually share a project with someone else just through an email address. So if I want to share this data with somebody, I just put their email in here, now they can access it. So that's what makes it really beneficial, especially for our Embry network. Okay, so GitHub. Who all is familiar with GitHub? Few? Sweet. So GitHub is kind of the repository where all of our free tools exist. And so I learned a lot about GitHub last year, pull requests, issues, all these different things. So what's nice about this is you can have a team of people working on a project and we submit into this repository that's online. It's freely accessible. All of our R-code, everything that is part of this package is in this repository. So this R folder, which you won't need to go into, this is the behind the scenes of the package. These are all the individual R scripts that we had to write to make this package work and add in all these different functions. So all the individual scripts are in the GitHub. And then we have man pages for the manuals. And so all of the functions that are included also have a help. And you can see more information about what this function is and how to run it. So, but in addition, we have a vignette or a tutorial that will actually walk you through each of the steps. So really, when you come to this page, get that one. You would come here, and at the bottom is the README file. For any GitHub, the README is going to tell you what the package is about and how to do the install. The installation is always really critical because that's my first hurdle with anything. It's just getting it into the system. And so because this is not on, most R packages are on a CRAN, and you can just do install.packages like this code here to install a package. GG tools, there's various, I think there's over 7,000 packages now in R because it's community led. But because this is in a GitHub repository, I'm not actually hosted on CRAN yet. We haven't made it to that level. I'm not that sophisticated yet. So we have to use DevTools, which is your development tools package, to install it directly from my repository. And so this would be how you're going to install it onto your local machine. Once you have R installed, I use RStudio, which is what I'm going to show you, have those two things installed. And then the first step is run this line and run that line. And we'll walk through that. Once all of that is installed, that's kind of like hurdle number one. And then it's formatting your data to get into the package. And then after that, it should be pretty much just plug and play of the script. So every package in R, before you run it, you have to load it. So you only have to install it once, unless you change version somewhere or switch computers. You're going to do the install, it's going to be on your computer. Then the next time you come back to it, you do have to run the library ProteoDA to just load it back into that particular project that you're working on. And so what is included in this, see if I can blow this up. These are all the functions that we've actually added into this package. So the data import, the critical part you're going to have to learn how to figure out and format is have a data frame with protein intensities, your protein annotation. Annotation means like UniProt IDs, descriptions, gene symbols, any of that, any information that's describing the protein is your protein annotation. Metadata is going to be related to your samples. So which sample is the control, which one's the treated, do we have male, female, any of those other factors we talked about yesterday should all be in your sample metadata file. And so the critical component here, and I'll show you this, is the sample metadata file. Your samples are going to be listed as rows. Those row names of that file have to match the column names of your intensities. So that's how those two pieces of information get tied together. So you have sample one in the first column of your protein intensities. Sample one should be the first row in your metadata file. So those have to be in sync and tied together. That's what it's going to define all of your sample information. So once you have that, then we have a function that pulls it into this DA list. And that is just it's a S4 object that they call an R, where it's just a list of lists. So the list is going to include all the intensities, annotation, metadata. We're going to start putting in filtering, normalization, design model. Everything's going to get built into the same little object. If you have an error, it's going to be here more than likely because something is messed up in one of the files. And so usually there's like, if you're making a sample list and using a Windows machine, sometimes they add blanks or spaces or something like that. No spaces in R. The other thing when I see a lot when people are doing animal work in particular, their animals are numbered and they're not, R needs a letter. So if you have a number for your animal, you'll see I put like S in front of it for sample something just because I can't use a number in R. So what?
It can't start with.
Yeah, it has to do with the columns and the labeling and R. It needs to have a letter to start with so that it doesn't think it's a numeric factor. You don't want to make it a number. So that's got to be a factor. So there's different data types in R. You have factors, numbers, strings. So you have to tell your program what your characters are. So once we get in there, filtering, it's kind of a cyclic process. So we go through filtering. I'm usually filtering out, we run pool samples with projects, and I don't necessarily want to pull in the final results. So we can remove that group from the sample list before we do our normalization. If you've gone through your QC and you found an outlier sample, well, you can come back here, remove it with this filter samples function. We have one by annotation. So if there's like keratins you want to remove, you can remove those proteins from the list. So there's flexibility here in how you want to do your filtering. I also a general rule of thumb is 2/3 of the replicates in one sample group need to have a value. Because if I have 3 replicates and I only have one protein that has an intensity in one sample, I'm not doing statistics with one sample. So that's going to filter out those low abundant proteins that might have been identified, but not quite enough to be identified for statistics. That makes sense. So, yeah. Yeah. Oh, you're not on yet.
So I did have a question about that. Actually, I kind of the question was, I have seen statistics. I mean, the things that are on our volcano plots, but some of them did only get a single value for one, and it still takes the average and calls the others 0. Is that
Yep.
something different than what you're talking about?
No, that's what I'm talking. So it's an optional step. And so if you're really wanting to be stringent and you're just wanting your top 50, then you might remove like that bottom part of the proteins that has all the missing data, and you're just getting rid of those. But this is discovery, so I don't like to remove a lot of stuff. And so you leave it in, you're gonna have those zeros, and you're gonna see it on the volcano plot where it's way over here, because your fold change is now inflated because we have missing values. That's why I like our interactive plots, is to be able to interpret those. And I'll show you what I mean by that here in a minute. But yeah, so this is why you can't just hit a button and run the code, right? You got to make decisions throughout each of the different steps, and why they still keep me around, maybe. So there's options there. We try to design this where you really have a lot more power to do what you want to do with your data, and not us just being like, you have to do it this way. I don't want to make it so strict. But then the normalization is going to run my ProteoNorm app that we developed about 3 years ago now. So ProteoNorm took some of the functions that are in Normalizer DE package. There's 8 different normalization methods that are commonly used for proteomics data. And what we're doing is we're running all 8 of those methods, and then we're building a report. And the report will show how variable are they between your sample replicates, what's the correlation among your replicates. You want to have the method that's going to reduce the variability and increase the correlation. And we're talking about within a group, not across groups. And so we look at the report, we pick the best normalization method, and then we write the QC report. And the QC report is going to be your PCA plots, your dendrograms, that extra pieces of information. Once you look at the QC, that's when in that BIANN plot, you can see like, okay, I've got a lot of missing down here. I might want to do more filtering, or you can kind of start making some other decisions. So once you pre-processing is like probably 80% of bioinformatics job is just formatting and pre-processing. Once you've got the data cleaned up, the statistics, this is kind of what I talked about yesterday with the models and the adding the design. That's a little formula that you put in. Contrast are your comparisons. What do we want to look at? And then it runs the model. So after you've designed it, you told it, we just want to look at group, or we want to look at group plus batch. Then we run the model and then we extract all the results. And the results will, we have two functions. One, just to write all those Excel tables and files, the flat files for future, if you want to make heat maps or anything else, we have all the data tables. And then the plots is going to provide some static plots, but also that interactive HTML file. So, and that's generally what we send with every project if you use our core. So that all comes as part of our service. So that's kind of the high level overview of the package before I just like dive right into some R code. So yeah, there's this. All right, are we ready to go to R?
Can I ask another question?
Yeah.
So my quick question is, like, I trust you to do this a lot more than I trust myself to mess around with it. So why would I ever go in and want to do anything other than stare at the Excel files, as which you know I do?
Usually this is students, postdocs, somebody want to learn R, and you have a lot of different comparisons and you're wanting to like investigate something else, and I can't get back to you that fast. Yeah. So, but it is kind of nice to know like what's happening behind the scenes a little bit. So in the script, Dennis shared with everybody, there's some extra. So let me start up. This is RStudio. Anybody seen RStudio? Okay, we've got a few that haven't. So the way it works is there's different windows. At the bottom, I am in a directory, and this is going to show me all the files that are in where my files are in the directory. This top pane is my actual script. So my ProteoDA example R script is loaded here. The console is where the program actually runs. So that's where we're going to actually submit the code for the program to analyze the data. When it does that, over here is your environment, and it will show you what's been loaded in, what's happening as you go through the functions. So for a Windows machine, I had some errors with the install, which was this, the newer versions of R had this install lock turned to true, and it wouldn't allow me to install anything. So I've left some of this here as just, if you run into installation errors, that's what fixed mine specifically for Windows. Mac, I haven't had any issues yet, but I just thought that would be good to leave. So install packages, dev tools is that first step. Probably try to make this a little bigger. It's kind of hard to do with Zoom.
I don't know why this is so important to me, but I've gone through this before, and I just want you to know that when you're doing this and you pull this up, you really, what she's doing is everywhere that it's got one of those lines where it starts with a command that's in white there, she's hitting Shift and Enter.
No,
and then that
Control Enter.
I'm sorry, Control and Enter, and then it runs that.
Yes.
And so then all the lines that have the hashtags or whatever, those are like comments.
Yep.
Whatever. And so I'm sorry, that's where I had to start.
Well, that's
because I
where
kept
she's
seeing her do
going.
this hocus pocus, and I was like, I don't know what she's doing.
Get there.
Okay,
Because, yeah, we do this every day. It's kind of like, yeah, you just turn it on.
just do it. Yeah.
But yeah, so the dev tools is what y'all need first. And so that cursor can be anywhere in the line on this row 15. So if you have the cursor anywhere and then you hit Command Enter, Control Enter, like you see here, it'll actually send that line down to the console. Now we know, now you can see that I loaded up my package because I already have it installed. I don't need this. Another problem you might run into is if you have an error here is building the vignette. I have seen some people have issues with that. Just turn that to false instead of true, and that should fix it. And you can always email me if you have trouble. Glad to help get you going. So don't feel like this is like your last opportunity. So Once you have that in, if you built the vignette, this line is your access to the tutorial. So hit Command Enter, Control Enter, takes you to a web browser. I use the HTML option, and now here's the full tutorial. I believe this is also in the GitHub that you can download, but here I'm explaining the different data files you need, how to fix them, how to run all the different functions, what they do. There's just a lot more explanation that we're not going to read right now. So that is available as well. So I'm going to close this. And I have something else there to show you in a minute. But all right, so like I said, the hardest part is really getting your data put into the system itself. So the package itself includes an example data file, which is this code here that you're going to have. Whenever you're doing R, whatever is on the left side of this little arrow is going to be what you're defining you're defining that particular term. So let me just run this so you can see, hit Control Enter. So now you can see that on our environment here, we actually have a data, something popped up and changed. If I click on this, this is my full data matrix, my full table of all of my intensity and protein annotation table. So everything is in one file. I told you you needed 2 files, but in this case, I'm loading it in as one file that has UniProt ID, protein information. This comes from the FASTA header. Any other information, molecular weights are in this one. And then we start with the actual samples. So I have the first 3 are the pooled samples, and then it starts with sample 1 all the way to the end. So these are going to be all of my samples for the project we want to analyze with their MS intensity values. They're not normalized, just raw data.
Can I ask you a quick question?
Yes.
Are you setting the working directory, or where are you
Yeah.
saving these to on your computer?
Yeah, so right now, wherever I opened up that R script is where my working directory is, but yeah. So if you do Git working directory, this is where I currently am, is in the workshop ProteoDA. If you want to change the location of where your files are or where to save them, you change this to set WD, open that parentheses, and then you have to put your path in quotes. And so for Windows, it's different. For Mac, you're doing like this, but for Windows, things are different. So I'll give you an example. Like I'll send it to you because the slashes get backwards between Windows directories and Linux because we can't keep anything the same. God, everything is dependent on your environment. So this is also the reason why we turn this into a package. It works really well, just regular R scripts. It's fine if you have everything installed on that machine and you never change versions and no other package ever updates, then you're good. But once another package updates, everything's broken and it won't run anymore. So by turning it into a package, it's more self-contained, and all of those versions and dependencies are built in together, makes the install process much better. This took us a year to take all those scripts and turn it into an actual package. So yeah, it was a team effort for sure.
That would be something we come back to you y'all update it, and then we come back to this.
Yeah, so if you go to like this is where the GitHub is super powerful for interacting with the community. So say you have an issue, you run into an error, you can go to my GitHub page under issues and you can create this new issue. And so you'll see people here, I had errors with this or I had errors with that. And then we can go in and either fix the error or tell you like, this is what you need to do to fix it. So it's still interactive with the community and builds that package. So this is just version 1. I'm sure we'll add more functions and other things later. And then you get version 2.0. And so versions become very, very important to keep up with. Yeah.
So I've done GitHub before on a different program, and what you've got here is so much more user friendly. So thank you.
Yeah.
But I need to go farther back.
Okay.
What is your recommendation for how to learn R? Like, I
Yes,
mean, when
yes.
you're doing this
Yeah.
control, right? I mean,
Yeah.
I watch my kids do this
Yep.
and they're like, oh, when I'm here, it works this way. And I'm like, whoa, so, the hidden stuff, where should I start?
So a lot of that is on Stack Overflow, BioStars. Is like if there's a function you're looking to try to do, Google is your best friend. Trying to make that search term as specific as you can. I do have another notebook that I'll show you that I created for the sandbox because I was forced to. But anyway, that one I try to go more base R code. So instead of using these functions that you have no idea what the function is doing, I try to break it down. How do you actually make a box plot and stuff? So we might as well just go there now. We'll get back to here. So this is the, I try to add in like kind of what are the basic steps for protein analysis. How familiar are people with the NIH sandbox training module that's been going on? A little bit? Okay, we got a few. So NIH last year provided supplements to different embrace in order to come up with these training modules to host them in their own NIGMS sandbox GitHub. That GitHub has 12 different modules. Some are ATAC-seq. We've got some machine learning. There's different topics that are built into that GitHub. Mine is a little different from the others because I was a pilot award before the supplement, so I didn't have to follow the same standard they did. So mine's just what I came up with. But again, we're trying to go through the import, these different steps. But what I try to do is also add in some terms. So we have a lot of mass spec terms that we talk about: TMT, max quant, multi-notch, DDA, DIA, just little brief definitions in here, missing values, batching effects. How do we do pre-processing, multiple different steps? ProteoNorm is, again, my other tool. It's also in ProteoDA. I did a little plug there. But these are different definitions of standard deviation, the median absolute deviation, all these various plots that are in our normalization. The methods that we're using to evaluate normalization is pool variance, intergroup correlation. BSN is another normalization method, cyclic OS, kind of our top two. So point being, there's a lot of information in here on about kind of reading up on them. But then as you go, I start here with installing packages. So these are all base R packages. If you go to the sandbox, you can actually just hit the play button and it would run. I'm running this one right now on my local laptop right here. So get everything installed. These are the individual. So if you don't load in my package, you actually have to install all of these libraries separately. You have to load all those libraries separately into R. Once those come in, you can change color schemes. So there's a lot here. Yeah, so I did kind of plug in a little DA, but I want to get to the plot stuff. So let's see, through everything. So I try to show like how when you read in a file, that's the first step you're going to do. Your first step is read CSV file. See if I make that without losing everything. Yeah. And so read CSV is a base function of R that we're telling it point to this directory. So everything that's in the parentheses, this is the name of my file that I wanted to read in from. And the working directory is important because if I don't tell it the full path, say this is in documents, folder, projects, whatever, if you don't give it the full path, it's going to try to look for this wherever you're currently sitting. So I have to pull that in, and then head command will let you view that in R, and it will actually show you the data table. So there's a lot here. Box plots, so try to put a little definition of what those are. How do we create a box plot? All this code is here. And the good thing with that sandbox, you can actually change this code, make little changes, see what it does to the plots and the features. And so if you break it, it's fine. This is kind of, I've got like the Pearson correlation stuff in here. Principal component analysis are not easy. So all of that is in here, how to plot a PCA. If I want to plot it and color it by group or color it by batch and different kind of plotting functions. So it's all in here as well. I'll send you a link to that. It's kind of. But R in general, once you kind of start, and I think once you start playing through like a script that's already made, it starts to become a little bit easier to figure out what's happening. Okay, so far, right now, all I've done is input my data. And now we want to grab the sample metadata file. So one thing in coding, you want to make sure whatever you're defining these things as is short. You don't need it to be a long phrase, but descriptive enough to know what it is. So there's kind of a fine art to naming things also. So sample metadata file, I have 17 observations, 17 samples. 3 of those are the pools. And I kind of did a little trick in my meta file. I put a new column and I labeled it data column name. This is because this here should match the same columns that are in my input. So I have pool sample 1 MZML because that's the raw file converted to MZMLs. Well, this has to be in the same order as my data. And so we're going to define this pooled sample as a pooled sample from batch 1, sample name P1, just for pool. So then sample 1 is going to also be here, and that's our normal control. So that's the part that's really, really important. It's just making sure your sample list and your data names and the order of your samples match. Because if they don't, then you've labeled something control when it's really a treated, and yeah, your results are wrong. So, and you'll see here I added a S because the sample name itself really should not be a number. It needs to have a letter in front of it to make that a factor. So if I had gender effects or any other condition, I would just add that as another column in your metadata file. And in the code, these, whether it's capital or lowercase, is very specific as well. You've got to make that very specific in your code. So you can change it. You just need to know what you're doing to change it. So, okay. So now we have things that are in here. So this is a common R function called subsetting. So we want to subset that one file that now has the intensity data, has the annotation data, and I need to make that 2 separate tables so we can have 2 different lists go into our object, right? And so in order to do this, on my input data, the very first 4 columns is all the protein annotation data. So I'm going to call everything after those 4 columns is going to be my intensity data. So intensity data is going to equal my input data frame. I have the input data here. So I'm pointing to that table, not the metadata list. And then when you have the square brackets, whatever's in front of this little comma here, hard to see, these are going to be defining the rows. And so if you don't have anything there, it's going to take all of the rows in your data, in your file. And then, so it's rows, comma, columns. And then the columns, we're going to start with column 5 to 21. You can actually, and I've updated this in some other code, instead of doing 21, we do in call input data. So that kind of takes everything from that point from 5 to the end. And the thing with R2, when you have a table in here and say like here, I haven't finished typing out data and input data is popping up, you can just tab over and it fills it in for you. So that's also what I love about programming. We're lazy and we want to use tab a lot. It also keeps you from making errors. So before I break this, because I'm on a live demo and I probably will, let's do that. Oops. Like that, because doing this on the fly is never a good thing. Okay, so what we did there is we created a new table. So now I have 3 tables and one that has intensity. And you'll see this one had 21 columns and that one now has 17. So we got rid of everything, but we also got rid of all of our protein IDs. So now it's just 1 through whatever, and we don't want to lose our protein IDs. So we have to put all that back together. So we're going to grab the annotation out of that same file, grab the first 4 columns, make a new annotation table. We have the annotation that now defines. So now this row, this protein ID defines row 1 in our intensity data frame. So those 2 things are linked. And then I keep saying the row names of the sample metadata have to match the column names. So if we look at the row names of our sample metadata, when you load it into R, it's just going to be numbers of the different rows. We want to define those row names. And so we make the row names of the sample metadata equal to that data column name that I added into the R. So here, a quick way to do this in our function, I should show you this. So we start typing sample meta, sample whatever, and then I'm choosing the metadata, I'm tabbing over. And then if you hit the $, that's a scalar in old programming languages. And so that will show you the different columns that are in that particular table. And so you pull up sample metadata, I actually want the sample, that column to define my row names. So again, I'm not like typing that all out. I'm just using some tricks of linking to the table. I don't think I ever showed you that, Dennis.
Oh,
Yeah, so another little R trick. All right, so once you have that, now you have your 3 input files. I saw that. So we have our intensities, we have our annotation, and we have our metadata file. These are the names of what we just created. So when we're doing this DA list, if you name it something else and you don't name it this particular thing, you'll have to change this in this function to whatever you named it. So that's what I'm saying, you have to change things. It's, if you made it a capital instead of lowercase, you'd have to make this a capital I. So those things are going to feed into those tables, and we're going to create our first DA list. This, like I say, if you have an error in your file somewhere, you're probably going to get a red message down there on the bottom, and then we'll have to kind of figure out what it's complaining about. So right now I've got everything into my DA list. So instead of the individual files, we now have this object. And this object now takes all 3 of that piece of data and adds a list of a list and a list. And we have 3 null categories here, and that's all going to get filled in as it goes through the functions. Everybody lost? Perfect. As you should be. Yeah. Why do I have a red mark here?
It's job security, right?
I know. I like it. Where are you? Okay, so our next step here is we're filtering out samples. So again, when I do this analysis, I'm not interested in comparing pooled samples to my normals. So I don't want to normalize those in my data set because that's not what's going to be in the comparison. So we would, if you want to look at pools, you can keep it in and do your QC plots and see how they go. So we do evaluate the pools, but not in the final analysis. So this will do filter samples, taking that raw DA object that we created, pull out that data frame, select from our group category the sample that says pool. And that pool is specific to that metadata information. So based off of this group name. So if I made that a lowercase p in my sample metadata, then my R-code needs to be lowercase pool. So everything that I've kind of got highlighted in green is something that is related to your input file. how you defined it. And don't remember green because that's the color version I have in my RStudio. When you pop up RStudio the first time, it's probably all going to be white and not this color. But anyway. Like, okay, so we got rid of the pools. Ugh, get rid of that. Eek. Just trying to make this where I can kind of make that little bigger there. Okay, so the other key point, and this relates to how we deal with your fold change in the Lima model later. So we have a function called 0 to missing. Right now, my data that I put in, anything that has a missing value is coded as a 0. Lima can omit zeros if it's flagged as an NA instead of a 0. So here I'm changing anything that's a 0 to an NA value. And this becomes a little bit of an issue which we need to add to it. Other programs use NAN. We don't have it where it'll recognize NAN. It's got to be NA. So 0 to missing, that's just going to change our intensity values that have zeros to now instead of having zeros, we should have, well, I'm on the wrong one. That's our raw data. So see if I can find any zeros in here, usually at the bottom. Yeah, and I won't find one now because I'm trying to look for one. Anyway, that 0 gets turned to an NA. And you'll notice when I do this, I didn't make this name raw again. I actually made a new object. I made a new filtered samples object. So I still have my original object called raw, but now I have a new filtered samples object. So that way, if I mess something up, I can go back to my raw one. I don't have to start all the way over. It's more of a safety thing for me. And so the 2/3 rule kind of comes in here with the filtering of proteins. So the minimum number of replicates, again, if I had 3 replicates, I would set this to 2. So I want to have kind of 2/3 of replicates within one sample group to have a value. And that's just kind of you're going to have to kind of play and see, do you want to keep some of these at your level of how much missing data do you want to allow into your analysis? And we're doing this based on the group. So the minimal group is going to be based on that sample metadata list where I labeled it as group, which is, look at that table, it's this one. So it's lowercase g for group. I tend to make anything that's going to be your main effect, your main thing that you're wanting to compare, and we label that as your group. Okay. So filtering proteins, and when you do this, it'll give you a little note at the bottom here, how many proteins were filtered and how many are left. So by doing that, I removed 203 proteins, and I still have over almost 9,000 proteins left in this data set for analysis. Okay, now we're going to write our report. And so again, you have some options. What's kind of here, the data frame I'm pulling from now is my filtered proteins object instead of the raw object. So we've removed samples, we've removed proteins. The grouping column is for the calculating of the variance between the replicates. So again, we're using group. Output directory, you can change this to save the plots in a different folder. So I'd label it the first folder, QC report, and then this is the name of the file that it's going to generate. So those are all changeable. Want to be careful with the overwrite function. If you were to save this and then you were to run it again, and I've left this on true, then it's going to overwrite what I already have. If you change it to false, it'll give you a warning and say, yeah. So that'll start some normalizations, given the file name. So yeah, I don't want to overwrite it because I already have a report. So if I click here, click on my normalization report, open it up, you can actually see the results from that normalization. And so these are the top 3 are all about variability. Each dot is the sample group. So this example only has 2 groups included. And so you can the log 2 is really the unnormalized data. So you can see how normalization is going to reduce that variability within your populations than if we don't normalize, if we just log 2 transform. So we have 3 measures of variability, then we have correlation among the replicates. And then this is a log 2 ratio where it's doing a lot of the fold change calculations. And so ideally, this data set does not look nearly as clean as what most of them are going to look like. This one's got some humps here. It's got some different populations of proteins here. This is formalin fixed paraffin embedded tissues samples. So they have a lot more variability, more things that may generate some different levels of protein. But what I'm looking for on this ratio is the center of that fold change, the density should be around 0, where you're not shifting it to a positive fold change or negative fold change just by normalizing the data. So typically for most projects, cyclic OS is the default if they all look pretty much the same. For phospho and lower abundant ones, VSN tends to perform better. So those are typically my go-tos. We do have the mean difference plots also included. So you want the orange line to flatline to become a 0 slope for the different methods. So this top one is the unnormalized log 2. This is the median. So these are a little different than your regular MD plots in that they have all the different pairwise comparisons. So if I had multiple groups, it would do multiple comparison type values, but it's just kind of nice to see the overall spread. One thing that we're concerned with is you don't want to have, just because a protein is really high abundant, that influencing our differences. So that's part of kind of what normalization is trying to do. Oh, okay, let's get rid of this. So you get your report. Then once you have your report, now we want to normalize our data. So again, I'm making a new object for my normalized data. The normalization method here, I've listed them above the code. These are your options to choose from. So again, we shorthanded cyclic OS. Instead of having a space between our words and really long, we made it short. So run that. Now we have normalized data, and now we write our QC report in a very similar fashion, except I'm going to make this false because I don't want it overriding. I've already written my report. And these graphs kind of get updated over time too. But we've got all of our, the violins, kind of hard to see when you're sharing on a big projector, but down here is going to be those low abundant missing value stuff. So sometimes there can be a really big density of missing value for like DDA projects where we did gels or just injecting samples independently. We get a lot more here. So we still have projects that are label free and without the DIA approach. And those will include a lot more missing. And so there's more you got to deal with on missing values. But PCA, here you'll see we've got 2 samples that are kind of looking like outliers. And they're kind of looking like outliers in our cluster dendrogram. So 2 measures looking at similarity, showing that the same samples are really kind of different from the others. This is where you would kind of look at what happened to these samples. Do we have a reason to throw these out? Those would potentially be our outliers that we want to go back and remove from the analysis. So there's that. And then I've got the just correlation again of replicates. I don't really use that one. But the missing value one is again kind of helping us evaluate if we need to be more stringent with our filtering, less stringent with our filtering, those types of things. So you got some control there. This plot is only showing the proteins that have a missing value. So this is not the entire proteome. If I do that, it tends to break. It can be at 8,000 or whatever, but yeah.
Just curious, what happens, let's say you have 4 replicates or something and 3 give something, a value that's very consistent and 1 is just missing. Rather than taking the average, is it can you tell it to just ignore the
Yeah,
one that's?
so that's when you turn that to an NA value, then Lima should omit it from the calculation. It shouldn't go into the full change. If you leave it at 0, then it gets averaged in and it'll run it with that calculation. So the only time I would actually maybe consider making something a 0 is if we know it's a true knockout and we have reason that that protein's knocked out. So we can say that that's a 0 and calculate a full change. But that's with evidence and biological evidence that it's actually not there. Yeah. So a lot of it becomes kind of how do we interpret the end result? Yeah. All right. So the design part that I was trying to, that's probably the most challenging part of all of this is setting up this design formula for your project. And that's where I'd really highly recommend you can just search Charity Law, Lima Guide. She's got all the models. I mean, it's a really good paper and I have to read it a lot because I get lost. But but this is what you're looking for. So we're doing the no intercept model, which means I have this tilde 0 plus group. To run a intercept model, you actually just do the tilde group like that. And what that does is it's gonna, if I do it with the intercept model, I can't define the order and the way I want to do my full change calculations. So I like to do this one so I can actually define my comparisons and put that in. Say you added a sex factor, there's male and female in here. In your metadata file, you should have a term labeled sex, gender, whatever that's going to be. And then you would add that here into that model. And it's going to then take that into the model estimates and account for that difference. So batch, this is also how you would add in the batch factor and try to adjust for that. Paired samples are a little different. They're a little more complicated. And I think I have examples for that. So if you have questions about any of the functions, and when I mean functions, I mean this add design. That is a function of this package we wrote. So there's code behind there that we wrote for that function. This is the data frame it's going to apply the function to. And then this is the design formula is part of that function. So in R at the bottom, you write question mark, start typing the function name. A bunch of different functions come up. Choose the one you want. I just arrowed down, press enter. So now I've got question mark, add design, hit enter. And now we have a little man page that pops up over here. So that's where you can get to help information within R itself. And so here's the description, the usage. So all R functions, this is the function we're using. It tells you you need to choose a DA list object, whatever your raw, your filter, whatever we named it. And then the function design formula, you set that equal to something. So this defines DA list is this argument. Design formula tells you it's a string for the design matrix using intercept, no intercept, additive, all these different model types. And then we have examples because I like examples. And so in here, try to say this is the intercept model, how you'd set that up. We're using this no intercept model, but if you had an additive effect, which is what we're doing for batch, sex, to remove any of that, that's an additive model. Is putting that into here. If it's paired gets tricky because pairing, it actually generates a correlation matrix first, and then it does the modeling. So there's a lot more programming you have to do to add in that repeated measure into the sample. So if we just add your paired sample in here as an extra factor like we did batch, it's going to make that one I'm kind of getting into some statistics we probably don't care about, but there's mixed effects and random effects. And so it matters how you do it, whether you're just adjusting it out versus you're going to calculate this correlation of that repeated measure and deal with that repeated measure paired sample. So pairing is a different model for sure. which kind of looks, yeah. So I have fixed effects, mixed effects. We're fixing in the paired sample. Here, we're making it just some random factor that's included. So fixed plus random effects. It hurts my head. But you need to be aware of it because we run the models wrong and then it becomes not the correct interpretation. All right, we're almost there. And then we can almost have a break. All right, so I did my design. So we've added it. Well, actually, I don't know that I did. I did not run my design. So if you don't know what function you ran last, if you click down here on the console and hit the up arrow, it will cycle through your previous commands. So if you want to rerun something, you just hit the up arrow. All right, so I need to actually run this. Generate our design and add our contrast. So now if I go to my no intercept DA list, I actually have something in this design that was before it was empty. So now the design formula is here, the design matrix, I've added in my contrast. All of this is now filling out that DA object. That's why we made the object in the very beginning. This is important because we typically actually, instead of using a contrast vector and then defining my contrast here within the R-code itself, you can make a contrast file. So we have another function, contrast file, and then just point it to the contrast. So I usually name it contrast CSV. But what's within that contrast file is literally just on the first, A1 of your Excel spreadsheet is this phrase. So cancer versus normal is going to be the identifier that labels your Excel files. That's what labels your volcano plots and labels the HTML. So whatever you want that to say on those reports, you would put on the left side that equals. On the right side, cancer versus normal, these are very specific to your metadata file. However you defined your groups in your original input file, this has to match because that's what R's looking for. And we have had issues several times. If you're, like I say, if you use like a Windows machine, you pull up Excel, sometimes they'll add a space at the end of a group name and then it won't be able to read it. So you don't want any spaces in your files. I got to remove this or I'm going to break it because I don't have an actual file named contrast. Let's get rid of that. And that. A little bit more. Sorry. There we go. Okay, so we've added our contrast and now the lemma's going to do its work. Lemma has will fit the model based off that. And then the other thing to pay attention to is in the results. The p-value threshold, again, this is what's going to color our volcano plots. So if you don't want to use a 0.05 p-value, you can change it to 0.1, whatever you want to do. That's what you're going to color. 0.05 is the standard. Log fold change threshold is it's log 2. So a value of 1 is 2-fold. If you want to go down to 1.5 fold change, then that would be like 0.58. So just know that that's log 2 scale that we're looking at. Lemma also has multiple different methods for adjusting your p-values, like Monferroni and various ones. Benjamin Hochberg is the most common used. That's your FDR adjusted p-value. And so that will generate our results. And then the part we've all been waiting for all day long is to get our tables and our plots. And so once we have our results, we write the tables. Again, you can make another folder for the output directory. Yeah, I don't care about any changes there. The one that's going to matter is the plotting function. So let's go back to our protein annotation data. So in our annotate, and this changes by program. So like MaxQuant has a specific standard output. and Spectronaut has a different one. They have different labels for protein accession numbers and descriptions. So whatever you have in your annotation file, those 4 columns is what is going to be listed as the table column names. And that's what's going to pop up in your HTML file at the end. So whatever you want to see in that final report should go here, but it has to be very specific to that output. The title column is going to be the bar plot that, or the plot that shows up on the right-hand side. If you hover on a dot on the volcano plot, it pops up a new plot, and that's going to be the label for that one. It'll be better if I actually show you what this looks like. One thing, because I forgot to mention it earlier, another requirement of your intensity data going in at the very, very beginning, let me pull that up, my input data. You have to have a column that says uniprot_ID. That's the critical one that's going to link to all of your protein names and IDs. So it has to be all underscore letters, lowercase letters. Can't get my terms out anymore. But uniprot_ID. So that is also required. All right, we're going to get there. Okay, DA results. So I'm going to go to my folder so it's easier to see. I moved to a different location. All right, DA results. So we have several files. I know you can't see that. My laptop doesn't do it, but we have static plots that are just the PDFs of all the plots that are included in the interactive one. Going to the HTML. Okay, so lots in this file also. Let's make this bigger. So this is formalin paraffin embedded tissues, and actually, I think I do want to pull up this one to make that point. Yeah, so we provide a p-value histogram that is also in the static plots that probably no one ever looks at. But this will kind of give you indication about other confounding factors that might be present in your data set. When you see on the unadjusted p-value histogram that there's not a lot of proteins passing significance, and then the FDR adjustment is just going to flatline out, this structure of that histogram actually indicates there's another factor that's confounding these results. In this particular example, it has to do with the clinical factor in the samples that's not included in the model. And if I were to include that factor into the model, this would probably look a lot better and stuff. So yeah, sometimes we don't get all the information up front. And if we look at this, I can kind of go back in a post consult and say, what else is going on with these samples? And get a little more information and see if we can add that all in. And I do actually have a PowerPoint slide that we found on a blog that actually shows these, what that looks like. I'll give that to you later too. I'll add that to the thing. I just thought about it, sorry. All right. So in your plot, and you would actually get one of these with the IDEA logo and everything from our version. I didn't want to give everybody a IDEA logo on your own version. So the logo's removed. But you click on the dot. If I can get there. And so this is the protein that's going to be on the upregulated. If it were significant, it would be up in the cancer group compared to the normal. So the labeling is going to give you an indication of the order of the fold change and how we interpret it. So I do this on purpose so that I remember how we ran things. So anything that's on a positive upregulation is what is going to be up in that first group listed in the label. If you forget, you can click on it and then you can see these are our 2 groups in this data set. And overall, that one is slightly better, increased than the normal group. There's a lot of spread in this these data points, and that spread of the replicates and variability among the replicates is what's impacting our fold change and our p-values, obviously. So we're looking at the volcano plot with the adjusted p-values. We have an option to change this to the unadjusted p-values. And so now you can start to see, okay, what are some of those proteins that are significant before we adjust? And you can see those have a little less spread in them than this one down here. And so it's going to have a better p-value. But this is where you get into that, the fun part of the biology. What's happening here? What's going on? If you hover over each of these dots, it'll tell you which sample that is for that particular protein. Below is the full data table. So if there's a particular protein of interest you want to search for, you can just search for it and then click on that in the data table, and it'll highlight it in your volcano plot, which is over here and there. So if you're wanting to kind of investigate abundance, so we can change this to an MD plot instead of the volcano. And now we have the average intensity along the x-axis, the fold change is the y, and the coloring is the p-value. So 3 layers of information. And this is where serum, plasma type projects, those proteins tend to be way over here in the high abundant range versus like you want a little low abundant transcription factor, like where is it on this list? Are we getting lower into that range? So I like looking at these two when we're trying to kind of figure out where are we on the scale of abundance. All right, any questions about this? Because we'll probably have a post consult later.
Yeah, I think maybe taking questions this afternoon about this after people have had a chance to digest.
Yeah, I think we're ready for a break, aren't we?